Welcome to the pharmaphorum podcast. This is web editor Nicole Raleigh, and today I have with me Lew Bender, CEO of Intensity Therapeutics Inc, a leading biotech company with a mission to help cancer patients live longer, higher quality lives through intra-tumourally injected therapeutics that have high tumor dispersion and cell penetration properties. Welcome, Lew.
Well, Nicole, thank you very much for the opportunity to explain what Intensity Therapeutics does. Appreciate it.
So, for listeners curious, Intensity's clinical programs have demonstrated the ability of its therapeutics to kill tumours and elicit an adaptive immune response within days of injection, representing a novel approach to cancer cell death that holds the potential to shift the treatment paradigm even for cancers that do not respond to immunotherapy. But before we sink our teeth into our main topic, Lew, perhaps you could share with listeners a little about yourself and the journey that took you to doing what you do today at Intensity Therapeutics. I don't believe your background was in cancer research, for example.
Yes. Well, again, thank you, Nicole, for the opportunity to share this. My background is sort of like a random walk. That's, I think, the best background you can have, not really planned it out. I'm trained as a chemical engineer, and I think in terms of chemical engineering. I first worked in a materials business. I lived in Switzerland for 5 years, working on materials for biomedical applications, but not actual drugs and things. And then I came back to the United States, went to get a business degree, and then I started out in a company called Hemisphere, where I've spent 14 and a half years learning about the business of drug delivery. Now, drug delivery does not mean I had a truck license and a lorry, as you would say in the UK, and was delivering medicines to pharmacies. Drug delivery involves the science of getting the medicines into the body at the right place, at the right time, at the right concentration. So I helped to create this delivery technology. I was in charge of, lots of different departments, learned a lot about how to make drugs, how to develop drugs, how to clinically test drugs. I got involved in all aspects of it. And we helped actually to create a drug that is a $4 billion product that uses a delivery technology. The drug is an oral form of the GLP-1 molecule, and it is about a $4 billion product. And the company that bought the company I was working for is Novo, and they created this oral protein. And so using that, my knowledge of drug delivery, movement of molecules, about 13 and 14 years ago, I had this idea as immunotherapies were really coming into play, the PD-1 antibodies, the CTLA-4 antibodies. What if these are drugs that stimulate the immune response. What if we could get them to focus on the tumor by killing the tumor in a slower way to allow for the immunotherapies to read it and target it? That was my idea. And, here we are. And I'm a complete outsider. I thought about it as a complete engineer. Well, how do we kill the tumors we can see? And how do we kill the tumors that you can't see, don't even know they're there? You need a systemic component to treat systemic disease, right? So local therapy is great to killing the tumors. Radiation does that, but it doesn't set up any kind of systemic. So with this new technology, this delivery technology, we went through a 2-year development program where we screened a whole bunch of active therapeutic cytotoxic agents. And we, lo and behold, showed we could kill tumors. We dose differently. We don't dose based on your height and weight, which is typically the way it's done for most drugs that are given systemically. They're trying to poison the cancer without killing you. They don't always succeed when they give these drugs. So what we do now, as you noted, is that we can inject into the tumor. The tumor will now absorb the drug. We dose based on the size of the tumor. And the bigger the tumor, the more drug we give. We give it over multiple weeks, and we can really cause the tumor to die. And it dies, as we published jointly with the National Cancer Institute here in the States, We published a number of papers, and we show that this immune activation goes systemically. We presented a lot of our data. We've made it all the way to phase 3 trial in a very deadly cancer, sarcoma, and a phase 2 study it's a local therapy. We could possibly also use it locally in triple negative breast cancer. And so we're doing these 2 studies. But my walk was, I didn't decide to be a cancer researcher right out of college, go to med school, get a PhD or any of that. No, I went and got a business degree. I have a master's in chemical engineering, and I'm a really trained on a lot of drug development from the 14 years I spent at Hemisphere. But I thought about things differently. And when you think about things differently, you come hopefully up with a different solution, and you come up with hopefully a different solution that's actually going to work a lot better than the current solutions. And hopefully, with the data that we've generated so far and the excitement in 200 patients, and I've met many of the patients, hopefully in these phase 2 randomized controlled studies where we're adding our drug in breast cancer to the current standard of care, we can increase the percentage of women that have a dead tumor at the time of surgery. And if a breast cancer woman with triple negative breast cancer has a dead tumor at the time of surgery, and unfortunately only about 50% of the women get that outcome, 50 to 60% with current therapy, we can increase that. We can make a major impact because the ability of the body when there's a dead tumor to prevent the disease from coming back and all node negativity is much, much greater. And there's a significant delay to the disease coming back in women with breast cancer if they have a dead tumor at the time of surgery. So that's our objective. We'll have data next year, and we're excited about the results. And we've seen some very promising results in terms of the ability to kill the tumor 95% up front, and then hope the chemo and immunotherapies can add, and we can get a lot more women with a pathological complete response. So that's the goal in that study. So it's kind of, you asked me, what's the background? And I gave you a random walk background of my career, which explains how I got from where I was at the end of college to where I am today. Hopefully the zigzagging was there.
There was a lot of information, some zigzagging, but it was a random, let's call it a hike, Lou, and thank you for sharing. So let's unpack some of that for listeners now. Let's tunnel down into the science and look more closely, perhaps slowly, at some of Intensity's clinical programs. So at ASCO this year, Intensity presented a trials in progress poster outlining its phase 3 Invincible 3 clinical trial of INT 230/6 for the treatment of metastatic soft tissue sarcomas. So to start with, Lou, perhaps you can tell us more about that.
So that's an excellent question. I think, hike in a direct line about the science. So look, injecting tumors has been the long sought goal as long as there's been chemotherapy. It's 60 years of chemotherapy, 70 years of chemotherapy. Why can't you inject a tumor and kill it and get what I do without my delivery technology? Well, as I said, it's probably 20 times that this has been tried without success. So why? So I wanted to understand why. And so I started to delve into the physics of cancer and the physical nature of a tumor. So tumors are very dense. They have a lot of stromal things. They have fat, high fat content. The cells are, a layer of fat surrounding water. They're fat and water. And injecting 100% water in a very high density, high pressure, high fat environment is not going to work. There's physically a chemical problem associated with the microenvironment of a tumor and a water-based product. All drugs have to be water-based because the body doesn't take solvents very well or any other thing. So as I said, when I was in the drug delivery business, we worked on a way to make drugs soluble in fat and water at the same time, transiently, not covalently. This is just simply mixing our delivery agent, our cell penetration enhancing compound, which is 100% in 100% water solution, and it makes payloads soluble in fat and water at the same time. And so this new chemistry, which is the source of our patents, allows now for cancer cells to and tumors to absorb these very potent cytotoxic agents that are in INT 230-6, two of the gram for gram nastiest chemotherapeutics there are. When they go over the whole body, they cause a lot of off-target toxicity. But when they go into the tumor in our formulation, they saturate the tumor and they kill the tumor, and there's very little that escapes the tumor. We've understood what the right dose is for each size of tumor, all the way from 1 centimeter in diameter tumor up to 15 centimeters in diameter. And we can now kill the tumor. And as the tumor dies, the two drugs also have immune activating properties. So they're non-specific killing and immune activating. And so they train the dying cancer cells to attack the cancer cells, or they train the immune cells to attack the dying cancer cells, and then they can go out and help the cancer cells that we didn't inject in other tumors throughout the body. So it has a local killing power, immune activating power that goes around and, if not eliminating the unseen metastases and the uninjected tumors, certainly battling them a lot longer than would be done naturally. We are seeing uninjected tumors shrink, which is known as an abscopal effect, and local therapies like ablation have no abscopal effect, and radiation has a very poor, maybe 1 to 2% abscopal effect. What we know about, we're seeing 20% of patients have abscopal effects. What we know about, and we didn't track all the tumors in the body. We didn't track all the tumors. We didn't track any tumors under 1 centimeter in diameter. And so what we're seeing is, for the first time, a very, very high abscopal effect. We're seeing people with sarcoma in our phase 1/2 study living, apparently much longer than what you would have expected for that population. It's a very sick population that we tested. And so now we've moved into the phase 3. And, we are still learning in the phase 3 because we're dosing a lot more drug relative to the tumor burden than what we did in the phase 1, phase 2. And so this has given us some understanding as to how to even further optimize the program. So we've paused the study, and we are now redoing the protocol to allow for the learnings from the first 23 patients to play out. We're also, in need of a little bit more money before we can restart it. But ultimately, it's a good thing to now take the time to really understand the learnings from the first 23 patients, because we're now really killing these tumors. And when they die, they get bigger. They become cystic. They're not tumors anymore. They're cysts, and then they eventually drain. And we're killing a substantial amount of these tumors. So we believe that, along with the immune response, is the key now to a metastatic disease. It's very exciting. We've seen very good results in the phase 3. We presented at ASCO, a trial in progress, and obviously no data yet. But that study is going to take a couple more years, assuming we can get the funding in a relatively short period of time. That study will take, a couple more years to read out once we've completed enrollment.
Thank you for that, Lou. Exciting science indeed. And hopefully that funding is not far off. So let's move now to the data on triple negative breast cancer. And it was announced last month that the first 2 patients receiving INT 230-6 achieved high levels of necrosis after 8 days in the phase 2 Invincible 4 study before they initiated the standard of care regime. Can you tell us a bit more about that?
So, look, as I said, we kill tumors. Obviously, what a woman is trying to do when she has a localized tumor in the breast and in these triple negative breast cancer is a very aggressive form because it has no proteins to attack. If it's ER positive or HER2 positive or any of these, you can use other agents. But in the triple negative, it's very, very aggressive. When it's diagnosed, it's usually diagnosed in the stage 2 or stage 3 phase. 62% of patients are diagnosed in later stages than stage 1. And unfortunately, when it comes back very aggressive. And what the research has shown prior to my getting involved in this is that if a woman goes through this brutal regimen of immunotherapy and chemotherapy, from which 0.5% will die before they even get to the surgery. If they go through that and they do achieve a dead tumor at the time of surgery, the risk of cancer coming back, it's called a PAT-CR, is only 16% at 5 years. Whereas if you don't achieve a pathological complete response, meaning a dead tumor at the time of surgery, it's 50-50 that it'll come back in 5 years. And it really comes back very quickly, starting in about 1 year, even earlier. It's almost a straight line down to about 50% over 5 years. So it's very important that women have better tools to achieve a PAT-CR. In the larger tumors, we believe it's less than 60% will get a PAT-CR, a 0.5% will die. And by killing the tumor from the way we do it, we inject the needle into the tumor in the center and the drug moves out. Our drug kills from the inside going out. Chemo, because the blood vessels are on the outside of the tumor, but breast cancer tumors are very poor in terms of the number of blood vessels inside the tumor. They have very low numbers of blood vessels. Chemo has to kill like peeling an onion from the outside going in. Killing outside going in, which is the current standard of care, and inside going out, which is our drug, hopefully will increase the percentage of women with a dead tumor and no node positivity. And that's what we're testing now in the phase 2 study. It's being done in Europe, in Switzerland and France right now. So we're very excited because what we are showing on scans is after 8 days, if you look at the press release from June 11th in the morning, you can see a bright star-like tumor of about 3 centimeters before we treat it. And then 8 days later, it's 90% dark and necrotic and very lightly colored at the edge, which is where hopefully the chemo will come into play. So this is very, very interesting, I think, and very exciting because hopefully there can be a much higher percentage of women achieving their objectives. But at the same time, if we really do see a massive increase in pathological complete response, maybe we can get rid of some of these toxic drugs like anthracyclines. It's called the red devil, doxorubicin. It's called the red devil because it's a red formulation. And this is the drug that kills you and makes you like hair loss and weight loss and all this stuff. And if we can potentially get a regimen that has high PAT-CR, high dead tumor percentages without that drug, we can not only make it more effective, we can make a regimen that's more effective, we can make a safer regimen. And women won't have to fear the regimen as much as they fear the tumor itself. And that's kind of the goal in this first study. So 2 very exciting programs, metastatic disease to help people live longer, feel good. Our drug head-to-head against the standard of care. That's the sarcoma study. And then in breast cancer, our drug added to the standard of care. But if we see a real good rate, maybe we can start to remove some of the toxic agents that are in that regimen in the phase 3 studies. So that's 2 very exciting programs.
Exciting indeed, and interesting, as it was termed by you earlier. Now, if we stick with this notion of it comes to receiving a breast cancer diagnosis, and sort of the flip side of that fear being courage, I've heard that Intensity has entered into a collaboration to educate patients with early stage breast cancer about potential new therapeutic options. And this collaboration, for listeners interested, is with the model, author, executive producer, speaker, and breast cancer survivor Christine Handy. And it involves a film, Hello Beautiful, which is an adaptation of Handy's novel Walk Beside Me. And the film recently won the Golden Palm Award at the Beverly Hills Film Festival, and Handy herself also attended the American Society of Clinical Oncology Congress in Chicago this year. So going back to that word interesting, can you perhaps tell us more about this collaboration, Lew?
First of all, every cancer survivor is a remarkable individual to me because what women and men put themselves through to live, the desire to live, and they take a tremendous risk, right? But that's what they want to do. So to meet this dynamic person, Christine Handy, and she came and she's interested in what we're doing. She came to ASCO specifically. She saw us at the booth, at the presentation. I took her around. I showed her what the industry is, the tip of the spear, so to speak, for the cancer industrial complex as it is. And, she absorbed it. She understood it. She sees the potential for new treatments. And she's a real dynamic lady. I mean, just an incredible human being. If you read her book, Walk With Me, she, before she got diagnosed with breast cancer, she had, a very harsh treatment for a colon issue. This is all in the book. I don't want to be spoiler alert. Yeah, she lives, obviously, but she had that. She had a wrist problem that almost cost her her wrist and hand. Then she got breast cancer. She went on, adjuvant setting with the doxorubicin, and a remarkable lady. Wrote a book about it. Book was picked up by a producer and director team at Mystic Films. Made a movie, and the movie has done fantastic. It's not yet ready for distribution, but she's been going around, and she's been doing screenings in cities. She won the Golden Palm at Beverly Hills, as you mentioned. I think she's won a number of other awards in the field. And it's just a remarkable story, but it's not just a story about suffering. It's a story about love because she had angels and all her friends took care of her for her battles with all of these illnesses. I mean, it's a story of hope because with friends and with caregivers, we lose sight about caregivers in this whole thing. The patients are their caregivers, and their caregivers are the patients, and the caregivers suffer as much as the patients. And I've met many of my patients, but I've met from our studies, but I met their caregivers, and their caregivers tell as much of a story about suffering as the patients themselves. We must remember that. And so, as a CEO developing a drug, how do we help patients? How do we help their physicians? And how do we help the caregivers? How do we reduce the suffering? And that's what Intensity is trying to do. And that's what I think this drug will do if we can continue. It's a very challenging time in healthcare right now. But if we can remember to fight for the patients' caregivers, and of course the doctors who see their patients die over and over again or get the cancer to come back and all these other things, if we remember that, you can get through anything. And this is my experience. My experience is I did not start this company because I needed something to do. I started the company because I had friends dying from cancer. I had family dying from cancer. I had my grandmother died from cancer, and I saw how when my mother lost her at age 13, how that affected my mother for her whole life. I had a cousin when I was in my 20s. He died of esophageal cancer. I saw how it affected him. I've had friends die at cancer. They've left behind their daughters and their sons and their wives and their husbands and grandchildren that they'll never see grow up. So everything that Christine writes about and everything that we've done in our collaboration is what we're fighting for. And because everybody knows somebody who's had cancer, and everybody's known somebody. My sister had breast cancer. My sister-in-law's had breast cancer. It's ubiquitous. And we need new things like intratumoral delivery with our drug, which is nothing does what our drug does. We need these new ideas, and we need to break the sort of thought process of it's got to be coming out of an academic university or this kind of thing or that kind of thing. We need new ideas. That's what Intensity is. Different way. I invented this. I started the company in my basement. I did the chemistry in my basement to start off. We partnered with the National Cancer Institute. We've been, working on a skimpy budget, but the data speaks for itself. And when we have the data in a randomized controlled study, I think the world will wake up and see where we are. We've presented at major conferences, ASCO, the Society of Immunotherapy of Cancer, the San Antonio Breast Cancer Conference has had us as oral speaking. ASCO's had us as oral speaking. SITC's had us as oral speaking. The Connective Tissue, which is the Sarcoma Society, has had us as oral speaking. The medical community wants this, wants better solutions, and so far we've been accepted in to present. So it's been a tremendous experience. Happy I quit my job to do this and start this company. It's been a great experience. My neighbors and friends have put money in. People who don't even know me have put money in. And it's just been, I can't tell you how, words don't even express how, I wouldn't call it completely satisfactory, but how gratifying it is to know that other people care enough to help somebody that's not a cancer researcher, by training. But I am now. Pretty good at it now. That's the story. I mean, it is a story about patients. I did this for patients. I will make sure patients can get this drug by any means I can in order to continue the program that we've started a long time ago.
Thank you, Lew. And what a story indeed. Inspiring speaking with you. Thank you so much for your time today. It's been an absolute pleasure.
Thank you, Nicole. I appreciate it.
And that concludes another episode of the pharmaphorum podcast. You can find more information about this episode, including a download link and information about previous installments at pharmaphorum.com/podcasts. The pharmaphorum podcast is also available on Apple Podcasts, Spotify, Overcast, Pocket Casts, Podbean, and pretty much wherever else you download your other podcasts from. Just search for pharmaphorum and follow and hit subscribe. Of course, don't forget to visit our website itself, where you can sign up for daily and weekly news and analysis bulletins. And do follow us on Blue Sky and LinkedIn at pharmaphorum. That's all for now. Thank you for listening.